Yeah, I'd like to thank the committee for inviting me to speak today and share with you the information that our presenters provided yesterday. And I am sorry that I'm not who you expected, so hopefully I'll be able to make up for that. Yeah, I just wanted to kind of go through what we looked at yesterday in terms of the clean sheet framework, what that is a little bit, and what the ultimate goal of the workshop was, going into some of the details of the heritable hazards of smoking that were identified by our presenters yesterday, summarize those findings and provide some of the context and future needs, and yeah, look into some of the next steps in terms of research and policy. So this is a slightly dense slide, I apologize, but overall the somatic hazards of smoking are fairly well established, and they're what have been investigated the most in terms of cancer, lung disease, things like that. But at the same time, we do have mounting evidence that smoking may also induce germ cell effects, which have very different consequences from somatic cell effects in terms of when the effects manifest and what the types of effects are, and how the existing regulatory paradigms that we have to assess genotoxicity don't necessarily really well account for what actually happens in germ cells and the consequences of that, specifically due to the temporality of the phenotype from when the effect occurs to when it's seen, windows of sensitivity to germ cells that are specific to the phases of development and where those phases occur in the body, and when exposures can occur, and the combination of genome, epigenome, environmental interactions that can create a lot of effects. So the kind of questions that we wanted to talk about and try to answer were how do we approach risk assessment for germ cells, given the kind of epidemiological body of evidence we have from past exposures of all the people that have smoked before us and what we're seeing in terms of disease burden in the population today, and given animal model data in terms of what could happen in the future, and then what do we need to actually do about that in the present. We wanted to the committee members wanted to bring together different stakeholders, including researchers, policymakers, regulators, as well as some different interest groups, to have this kind of conversation and utilize the clean sheet framework to refine our thinking about how we need to go forward. And so the clean sheet framework itself was developed by a working group of HESI a couple of years ago, and there's a paper by Kerry, Dearfield that you can look at for more details on what the framework is. But basically, it highlighted the prevalence in genotoxicity testing for a long time that has a focus on high-level concentrations of effects and single yes-no endpoints, which may be more relevant for somatic cells and cancer burden, but not necessarily as relevant for germ cell exposures and all of the downstream effects on those. The goal is to more come up with a regulatory risk assessment framework that can better determine point of departures and look at dose response to understand adverse health effects due to germ cell exposures. And so this lovely graphic that I stole, just to give an overview of how germ cell exposures can affect current and future generations, because germ cell development has different processes and occur at different times in the male and female germline, but basically, from primordial germ cell development through maturation and then on to fertilization and the future offspring, there are many steps along the way where there could be different windows of sensitivity to different compounds and different processes that are happening in the cell, mitosis, meiosis, that can affect what the exposure is doing and what the phenotype is going to result in the offspring. So I just wanted to summarize some of the effects that the presenters provided to us yesterday from existing studies. And for the, no, that's okay. I provided at the bottom of these slides the names of the presenters, who provided the information and not necessarily the primary study authors. So just to be aware of that, and you can either speak with our presenters, most of whom are here today, or refer to the recording of the workshop, which I believe will be posted online for further information. But in terms of what the exposure to smoking can do directly to germ cells, there's evidence from rodent models that it can, and these are primarily looking at sperm due to the availability of sperm cells compared to oocytes in the body, but yeah, increase the formation of DNA adducts, increase DNA damage to sperm, increase certain mutations, increase chromosomal damage, and overall reduce sperm function and therefore fertility capacity. And we see some of these same effects as well in studies of humans with increased aneuploidy in sperm, increased chromosomal damage as well, and also reduced sperm function. And then, yeah, since we're kind of looking at multiple generations, because the direct effects in those sperm cells in the parents can have downstream effects in the future offspring, the presenters provided us with some effects in the F1 generation, and I'm starting with human effects that have been documented from epidemiological studies. So for both maternal and paternal exposure, you see lower sperm counts in the sons of exposed parents, as well as increased incidence of childhood cancers, as well as with paternal exposure, increased rate of congenital malformations, and increased incidence of asthma. And this one was pretty interesting because it was found that the effect for this incidence was much greater when the fathers were, the smoke exposure was occurring before the age of 15. So at this early adolescent stage, the exposure to smoke had a much greater effect of the incidence of asthma in the offspring compared to when the preconception exposure in the fathers was later and closer to the time of conception. So that once again points to some specific germ cell developmental effects in the parent and that window of exposure that can have different or more pronounced downstream effects in the offspring. And then for some F2 level effects that were shown, which were presented to some extent just earlier, there were from maternal grandmother exposure, there's been evidence of increased incidence of attention deficit and hyperactivity disorder and symptoms related to that. And then from both paternal and maternal grandmother exposure, the increased incidence of asthma once again. So that's both in the F1 and in the F2 generations. There's also studies in mice looking at F1 and F2. So I've just pulled out some of the studies that were presented on mice. And specifically for exposure to tobacco smoke, there's been observations of increased incidence of the inflammatory phenotype indicative of allergic issues or issues related to allergies, in addition to decreased lung function indicative of potential for asthma in humans. And then when looking at specifically nicotine alone and not the tobacco smoke mixture, there's evidence that there in the F2 generation is reversal learning deficit, which is a phenotype I believe linked with ADHD. So this graphic unfortunately I have the nicotine being dosed to the male grandparent, but I believe this was specifically with the female exposed grandparent. But basically showing that there are a diverse range of effects across different generations from the paternal or grandpaternal, parental or grandparental exposure to cigarette smoke and nicotine. And then further to get to some of the understanding of the mechanisms behind these effects that we're seeing and some of the molecular changes, further molecular changes, there's some evidence of epigenomic changes in rodents specifically, and that this was linked to the study that was looking at the ADHD phenotype in offspring. The changes in DNA methylation in the sperm of the directly exposed fathers, which was specifically linked to the promoter of the dopamine receptor, and they did see further changes in the offspring in terms of dopamine receptor expression of mRNA in the F1, I believe, and also in these epigenome function or changes, I'm focusing specifically on directly to the germ cells and not any of the somatic DNA methylation changes, just because there are also downstream effects that have been documented in the somatic tissues, but just as far as the immediate germ cell changes themselves, in sperm of directly exposed humans, there have been changes in DNA methylation as well, and the regions that have been differentially methylated have been linked with areas of retained histones, so it could indicate a link with histone to protamine transition in the sperm. So to kind of wrap up what we know at this point, there are several lines of evidence supporting cigarette smoke as a genotoxin in germ cells, and nicotine alone also appears to have effects which could have implications for your e-cigarettes and other smokeless tobacco products. And then the evidence that we have so far indicates that there's a combination of germ cell DNA and chromosomal damage, DNA mutations, and epigenetic alterations that can manifest in these observed effects. But we don't necessarily have a clear adverse outcome pathway established, and there could be differences with different exposures because the tobacco smoke itself is a very complex mixture. So kind of dissecting those pathways still needs to be done a little bit more. But overall, given the prevalence of exposure to cigarette smoke in the population and the potential for small effects in germ cells, changes to germ cells that can amplify up into larger effects in the future organisms and the population as a whole, there is a high level concern that needs more attention. As far as the data gaps that still need to be addressed, we don't necessarily have a lot of solid dose response studies, and to understand concentrations that will elicit the effects, and there's also, as I mentioned, a need to better understand mixture toxicity and the particular drivers of the effects that we see in germ cells, especially given in humans how we're not exposed to just one substance at any given time. We have a lot of co-exposures, and could there be various synergisms or different effects given other co-exposures? And further understanding the molecular pathways leading to the multi-generational effects is needed, as well as looking more into effects on the female germline and those specific mechanisms, since more of the information is focused on the male germ cells right now. And so further studies should definitely focus on or ensure that they're looking at appropriate windows of sensitivity and kind of understanding when when the exposure, germ cells may be more vulnerable to exposure, making sure to look at relevant endpoints and tissue types and sampling to get the mechanisms better understood, utilizing more of the high-resolution multi-omics data and integrating those to better understand changes in the phenotype, linking back to genome and epigenome changes, as well as accounting for the sex-specific differences that are seen both in the parents and in the offspring, and better understanding those. For future actions, we're interested in reconvening the workshop regularly to continue the conversation and try to share progress and ideas for future research, and try to provide some of this information to key regulators to promote further action and support further research, since we do feel like it is an urgent issue to be investigated further and potentially take regulatory action on. And also, in the meantime, develop communication strategies to provide the information we have to the general public to potentially help inform their understanding of what might be happening when germ cells are exposed, since it's not there's not much awareness in the general public regarding that. So I just wanted to thank all of the committee members and presenters from yesterday, and many of whom are here right now. So if you have detailed questions, you might want to direct them to the speakers instead. But I'm happy to answer what I can.